This first one, though, is going to give us some ideas about specifically what we're doing with DNA and how we use that DNA in various ways. The first thing we want to understand is how do we go about essentially collecting DNA and making that DNA so we can analyze it. And the way we do that is by making copies of that DNA in a synthetic situation. And that's through the process of polymerase chain reaction, PCR. And so we gather our sample, our genetic sample of interest, whether that's from a crime scene or we're just trying to identify the function of a gene. We want to identify paternity, whatever we're using it for. We're going to gather the sample and then we're going to stick it. And because the sample that we gather is going to be very, very small. Now there's various preparations you have to do, right? You're not going to just be able to like pull a few group of cells and it's going to mostly be DNA. You actually have to get rid of the membranes, and you have to cause the DNA to condense and various things. So it's a set of chemical reactions to get that to happen. But once we do that, we're then going to make copies of the DNA. And once we have copies of the DNA, right? To make those copies, we needed to understand how DNA replicates. And so that's one of the main things in this lesson. I believe I did a video on that, the DNA replication process. Yeah, so I won't go over that too much in detail, just to say that, right, that DNA is semi-conservative. So we have two strands of DNA that complement one another. So A pairs with T, G pairs with C. And when those two complementary DNA strands open and separate, we then have a few different enzymes come in that are involved in the process. But ultimately, we're going to build a complementary strand that matches this one, and this is our original here, and this is a complementary strand that matches this one. That's how we make copies of our DNA, by splitting it open, breaking those weak hydrogen bonds along the center, and then building copies of it on each side. To do that, there's a few things that are necessary. We have to have primers, and that's placed down by primase. We have to have the builder that links the nucleotides together to form the complementary new strands, and that is DNA polymerase. And we need something that kind of breaks the bonds open, and that's called helicase. And so once we understand how that works in regular DNA replication, what our cells do, we then can take that process and use it as the template for PCR. And so, well, we need to open up the two DNA strands, right? Helicase does that in DNA replication. We're going to use heat to do that. Now, it works because the bonds between the two DNA strands are weaker. We'll talk about the types of bonds later. Because those are weaker, they break at a lower temperature than the ones on the backbone would. And so that's how we can take advantage of that. So after we heat it up and split it open, we're going to cool it down just a bit to get the primers that we need to attach. Now, these primers are typically going to target, in the case of identification, those STRs, those regions that have repeats over and over and over again, because those repeats are highly variable from one person to the next. And that's what we really are interested in for trying to identify individuals, is areas of the DNA where there's more variation. And so the more of those short tandem repeats we have, the longer this segment of DNA will be for one person compared to another. And so that's why we target those STRs for those reasons. If we're working on genetic modification, we'd be targeting specifically our DNA of interest. And we can produce those primers synthetically, and that replaces the job of primase. Next, so we've split it open, we put the primers on. Now we need to have the DNA polymerase build the new strands. Now that's what happens in our cells, right? The DNA polymerase is going to come in and build the two complementary strands to our existing ones. But because of the heating that we have to do to split the DNA molecule open, the DNA polymerase that our cells have can't survive. And so we use the DNA polymerase of another organism, Taq polymerase, that's evolved to live in hot places. and it builds the DNA strands for us. And so you can see that template of the natural process of DNA replication is what we've used to come up with this artificial process. And you've probably heard more about PCR now. That's what a lot of like the COVID testing was, particularly early on when we didn't have our protein tests to look for antibodies. Now we were initially looking for that genetic material, and they would literally do PCR to your sample and get copies of the viral DNA is what they were after. All right. So a couple of things that we use DNA for, I mentioned, it talks about it in the video. It's to identify, solve a crime, right? If we can get a sample from a victim that doesn't belong to the victim, that's a good indication of whoever was with the victim recent to their or near to their death. And so that can tie people to crime scenes and things like that. Paternity, so our Jack and Jill story, right? This would have been helpful for that. And then modifying genes, detecting mutation, recombination. We've done a lot of that, where we take the gene from one organism and place it into another organism's genome. And that's how we produce a lot of the insulin that we use today. And we have bacteria make the protein using the code for human insulin. All right, so once we have copies of the DNA, then we need to visualize that DNA. And that's what this gel electrophoresis process is, right? We can put our sample into these wells, and DNA is negatively charged. So if we put a negative charge on this end and a positive charge on this end, we can pull that DNA through these wells and cause them to separate. Now, the thing that's going to happen is the longer the DNA strand is, the slower it's going to move through these wells. The shorter it is, the faster it's going to move. And so if we have lots of different fragments, lengths of DNA in there, we can separate them out, and it creates a pattern that we call the DNA fingerprint. And so if someone has more STRs, they're going to have a bigger fragment of DNA, and that DNA is going to move more slowly. And so this allows us to visualize differences in the individuals. Now we have to get those fragments, right? And we're not just going to, we're not going to just put like a whole chromosome in there. That would be terribly long. What we want to do is take the DNA and chop it up. And that's what restriction enzymes are for. And restriction enzymes are specifically looking for palindrome sequences. And that's the recognition site here. And this one is going to cut. And the reason why they're called palindrome is the GAA-TTC going 5' to 3' is the exact same going 5' to 3' down here, GAA-TTC. And so it's going to cut at that G and that G there, right? That's how it gets that separation. Now, so if we look at this strand of DNA right here, we can find the sequence GAA-TTC. That's going to be where our cut is, and it would cut there. Now, on a test or quiz, I'm not going to give you both strands of the DNA to see if you can recognize a cut site. I'll just give you one, and then you'll have to identify it. So that's the way that will work. We talked about the STRs. Those STRs are going to result in different restriction fragment lengths for different individuals. And that's what these RFLPs are. So, all right. And so now we can, our last question here, decide if it's possible that Donald is Jill's father. That means that any fragment of DNA that Jill has that is not explained by her mother, Daisy, must be explained by her father, Donald, if Donald is her father. So as we look here, okay, this length seems to be explained by both. This length not explained by Daisy, it must be explained by Donald if he's their father, and it is. Here's another one right here, not explained by Daisy, but is explained by Donald. And this one's not explained by Donald, but it is explained by Daisy. And so this would be a shortened example of the evidence pointing to Donald being Jill's father. We'd want to look at more sites, right? I think they typically will use over a dozen sites to kind of do this comparison and have enough statistical power to be conclusive. So